Hello everyone, welcome to MedBiotics. Today I'm going to talk about the ribonucleic acid, which is also known as RNA. So RNA is also made up of nucleotides, and each nucleotide contain pentose sugar, phosphate group, and the nitrogenous base. So let's look at these three things that I have just mentioned. So for pentose sugar in RNA, we have the ribose sugar. It also has 5 carbons, and the formula for ribose sugar is C5H10O5. The only difference between the ribose sugar and the deoxyribose sugar from DNA is that there is one extra oxygen in the second carbon in the ribose sugar. So now we know that the pentose sugar, which is present in RNA, has one extra oxygen in second carbon, and it's known as the ribose sugar. Let's look at the nitrogenous bases we have in RNA. So we have purines and pyrimidines. For purines, we have adenine and guanine, which are same as DNA. They both have 2 rings. Let's look at the pyrimidines we have in RNA. So we now know that in DNA we have thymine and cytosine as our pyrimidines, but in RNA we have uracil and cytosine. So basically we have uracil instead of thymine in RNA. So both uracil and cytosine have one ring. So this is the thing to remember about RNA, that we have uracil instead of thymine as our pyrimidine nitrogenous base in RNA. And this is the phosphate group that is present in RNA. So here is a structure of the RNA nucleotide, and we can see that we have phosphate group, ribose sugar, and the nitrogenous base. And we can see that the nitrogenous base joins the ribose sugar in the first carbon of the ribose sugar, and the phosphate group binds to the ribose sugar in the 5th carbon. So this makes a whole structure of a nucleotide of the RNA. So when one nucleotide binds to another RNA nucleotide to form the full single-stranded RNA structure, this ribose sugar here will bind to another phosphate group in this 3rd carbon position. So we can tell that the nitrogenous base binds to the 1st carbon of the ribose sugar, and the phosphate group binds to the 5th carbon and the 3rd carbon of the ribose sugar in a full single-stranded RNA structure. So let's look at the single-stranded RNA structure. So we can see that unlike DNA, for RNA there is a single strand, and it also goes from the 5' end to the 3' end, and the nitrogen bases which are present in RNA are uracil, cytosine, adenine, and guanine. Remember, uracil is only present in RNA. So let's look at the RNA structure in a bit more details. So here we can see that 2 ribose sugars from RNA are bound by this one phosphate group, and the bond that is created by this phosphate group here is known as the phosphodiester bond. So here we can see that this phosphate is bound to the 3rd carbon of this pentose sugar and the 5th carbon of this pentose sugar. So by the phosphodiester bond, these two ribose sugars are bound to each other in RNA. So now we know that RNA is a bit different to DNA. We know that as a nitrogenous base, we have uracil present in RNA, and there is an oxygen present in the 2nd carbon of the ribose sugar of RNA, and also RNA is a single-stranded RNA structure, but DNA has like a double-stranded structure, which we have seen in my previous video. Similarly, like DNA, RNA is also negatively charged because of the presence of this anion here in the phosphate group. RNA is negatively charged. I have written a bit more details about the phosphodiester bond in the description section of this video. I'm gonna make one more video which will describe the phosphodiester bond present in both RNA and DNA and how the bond is made and which reactions are behind this phosphodiester bond. Thank you so much for watching this video with me. I hope to come up with some new concepts and I will try to explain them as simply as possible. Till then, keep loving science and take care.